all, I'm going to discuss about the structure of astrocyte, then about the function of astrocyte, then about the method used for identification of astrocyte. So, the first of all, what's astrocytes? Astrocytes are star-shaped cells, okay, found in the brain, similarly to other neuronal cells. So, astrocytes comprise synapses or cell ends that allow chemical and electrical communication between cells. So, astrocyte also consists of dendrites that allow the communication from other cells to be transmitted within the cell body of the astrocyte. So the important function of astrocyte, the first of all is the structural support. So astrocyte provides structural support to neurons by forming a supportive framework within the central nervous system. They create a network of processes that surrounds and initiates the neurons, helping to maintain their position and organization. So the second function is regulation of extracellular environment. So this function is very important because astrocytes play an important role in the maintaining the chemical environment around the neurons. Okay, and the important is that regulate the concentration of ion, such as important ions such as potassium and calcium. So by taking them up from extracellular space and releasing them when needed, this activity helps maintain proper neuronal function and prevent buildup of harmful substances. Okay, toxic substances. So the important other function is nutrition or nutrient supply. Okay, so astrocytes are involved in uptake and storage of glucose, the primary energy source for neurons. So they ensure a steady supply of nutrients to neurons, helping to support their metabolic needs. So the other function is neurotransmitter uptake and recycling. So this function also very important because the astrocytes participate in the clearance of neurotransmitters from the synaptic cleft after neuronal signaling. So they take up neurotransmitters such as glutamate, and convert them to less toxic forms or recycle them for later use by neurons. So this process is essential for maintaining proper neurotransmitter balance and preventing excessive excitatory activity. So the other function is formation of blood-brain barrier, BBB. Astrocytes contribute and help to formation and maintain of blood-brain barrier and protective barrier that separate the CNS from the circulating blood. So they help to regulate the passage of substances between the blood and brain, protecting neuron from potentially harmful or toxic agents. Okay, so the other function is modulation of synaptic activity. So this function, astrocyte activity communicate with neurons and can influence the synaptic transmission, and they release chemical signals such as the glial transmitters that can modulate neuronal activity, synaptic strength, impacting information processing in the brain. So very important for modulation synaptic activity. The other function is the repair and regeneration. So by helping the astrocyte, astrocyte play an important role in their response to injury or diseases in the CNS. So they can undergo a creative process called astrogliosis, where they proliferate and form a scar tissue to isolate damaged areas and prevent further damage. So additionally, astrocytes release factors that promote tissue repair and regulation. So another part of this video, I'm going to talk about the important method used for astrocyte identification. So the first of all, immunohistochemistry, IHC. This technique involves antibodies that specifically bind to protein expressed by astrocyte. So by binding antibody to the specific protein, which is released or expressed by the astrocyte, we can identify the astrocyte. So antibodies agent markers such as the glial fibrillary acidic protein is called GFAP or S100 beta. So and glutamine synthesis, okay, and glutamine synthesis GS. So are commonly used to label and identify astrocytes in the brain tissue. Okay, so the other important method is immunofluorescence, like IHC. So immunofluorescence use fluorescently labeled antibodies to visualize astrocytes. This technique allows for identification or detection of multiple markers simultaneously, providing more detailed information about astrocyte subtypes or functional status. So in this, we can just identify that in the second method. It's the same as first method, but in second method, we are using the fluorescence. Don't forget that. So sometimes we are just mixing the method for more clearly and more easily to find out the structure of astrocyte. So the other method is transgenic mouse model. So researchers have developed the new technique nowadays, genetically modified mice, in which astrocytes express specific fluorescent protein, such as a green fluorescence protein, GFP. So astrocytes can be easily identified and visualized using these mice under fluorescence microscopy. This is very important for microscopy. For example, we can use confocal microscopy. So the other method is calcium imaging. So astrocyte exhibit calcium-dependent signaling. Okay, let's mark it. Calcium-dependent signaling. So and change in intracellular calcium levels and are commonly used as the functional indicator of astrocyte activation. So calcium-sensitive dyes. So we are using calcium-sensitive dyes such as Fluor4 or Fura2 can monitor calcium functions in astrocyte, allowing their identification and characterization. So here we can see the structure of astrocyte after identification in the confocal microscopy. See, this is all structure of astrocyte. So in this case, you have to calculate it, how many structure of astrocyte is present here. See, all this structure is the structure of astrocyte. You can identify it easily under the microscopy by using confocal microscopy, for example. So electronic microscopy, the technique provides ultra structural details of cells and their surrounding using electron microscopy. Astrocytes can be identified based on their characteristic morphology, including their numerous processes and close association with blood vessels and specialized cell junction. So the important thing is that to note that choose of identification method maybe is vary or different depend on the research. So depend on the research or experimental goals. So in addition, different technique offer different level of resolution, specificity, and information about the astrocyte function. And combination of method, as I told you before, the combination of these methods is often employed to obtain a comprehensive understanding of or clear understanding of astrocyte biology. Thank you very much for this video. I hope you have enjoyed and benefit from my videos. Thank you very much. Please don't forget to subscribe my channel and like this video. Thank you very much. Bye bye.